enzyme specificity, and there are three classifications. So specificity is really just selectivity, like how specific is each enzyme for a particular reactant or substrate. So absolute specificity means the enzyme accepts only one substrate. So something like urease only works on urea. That's it. So that's absolute specificity, only one specific substrate. Relative specificity means it'll work on like a class of groups, something that has similar functional groups, like perhaps all alcohols or something that has a similar type of structure. So relative specificity, um, you'll have a range of substrates, so more than one, but they have to be similar in some way. So alcohol dehydrogenase is going to work. We mentioned that earlier. That's going to work on alcohols, and it'll oxidize them to aldehydes. And then the last one is stereospecificity, where you're looking at it only works on a particular stereoisomer. So this is going to be very, very specific. So it reacts with or produces only one particular stereoisomer. So that could be like it only works on cis versus trans. It only works on D sugars instead of L sugars. So this particular one, only the product is what makes it stereospecific. It only makes the cis isomer. So let's see if we can apply these definitions to a particular case here. So the enzyme hexokinase catalyzes the transfer of a phosphate group from ATP, from ATP to a carbon 6, that's very specific, of a carbon 6, a carbon atom number 6 of D-glucose, D-fructose, or D-hexose. The L hexoses are not substrates. So right away you can tell that this only works on D but not L. That it's going to be stereospecific for sure, but there's more than one. So we have there, which of the following terms describe the specificity? So definitely stereospecific because it works on D but not L, D but not L. And then also it works on hexoses, right? Hexoses, so 6 carbon sugars. So it'll work on D-glucose, D-fructose, D-hexose. So that would be relative specificity. If it was absolute specificity, it would only work on like D-glucose or only work on D-fructose, something like that. But because it works on a whole group of substrates, then it's going to be relative specificity. And then because it works on D but not L, then it's stereospecific. Enzymes that bind several compounds that are similar in structure have what type of selectivity or specificity? So absolute, again, that would only work on one. Stereospecific is particular for one type of stereoisomer. So relative is going to work on um, on several compounds that have similar specificity, similar structure. So we mentioned that catalysts are used to lower the activation barrier for a reactant, and that catalysis isn't yep, enzyme results from an interaction between it and the substrate. So let's look more at the structures involved in enzymes. So enzymes are going to have an active site, that's the area where the reaction is going to happen. So it's the specific area of the enzyme where you're going to bind the substrate. And usually, so here's a picture down here. So usually this enzyme will have like some kind of, it's a cleft or a pocket. It's going to shield the substrate from the solvent, and that's going to help facilitate the catalysis. So that's the reaction that's happening. The enzyme can also align the portions of the substrate that need to react with the enzyme. That active site is going to be very important. And then it could also bind the cofactors, and we'll talk about the cofactors in the next section here. So what happens first? You have your enzyme, you have your substrate, they form an enzyme substrate complex. So there's going to be some kind of attraction between the substrate and the enzyme. The reaction happens, and then you release the enzyme and the product. So the enzyme should come out unharmed. The substrate should change. The substrate is going to change into the product. So that's your final result that you have there. So what is a cofactor? We mentioned a cofactor. Cofactors could be small inorganic ions, so it could be something like copper or iron or zinc. It could also be an organic compound. And if it's an organic compound, we call that a coenzyme. So coenzyme if it's an organic compound. But what are they? They're just something that's also required for the enzyme to perform the catalysis. So in order for catalytic activity of the enzyme to happen. Cofactors could maintain the protein conformation. It needed the substrate to bind and to form the activated complex. So it's something that stabilizes that enzyme substrate complex and enables the reaction to happen. And if you don't have those cofactors, if you don't, or the coenzymes, so if you don't have those organic compounds that are needed, or you don't have the ions, then the reaction's not going to happen. You could also have, so you need some of these enzymes, like or some of these ions. You do need some ions. If you have too many, or heavy metals that can sometimes be toxic. So there's a toxic effect of heavy metals. So cadmium and mercury can sometimes replace zinc. So zinc is a cofactor that you need. If you have toxic heavy metals in your system, they can sometimes replace the zinc in those cofactors, in those enzymes, and then they can inactivate the enzyme and then if your enzymes aren't working properly, then your body starts to shut down. So coenzymes are needed for specifically for oxidation and reduction and transfer reactions, group transfer reactions, but generally hydrolysis reactions, you don't need them. The proteins are able to carry those out by themselves. You also have some things called prosthetic groups, and this is a cofactor that is permanently associated with the enzyme in order for the enzyme to act. So a heme group, and we're going to see that again. We'll look at the heme group that has iron in it, and it's this big kind of complicated structure. That's going to be needed for cytochrome C, for hemoglobin, myoglobin. They're all going to need that heme group there. If the um, if the cofactor, the enzyme cofactor complex is there. So if you do have that prosthetic group, you do have the cofactor there, it's called the haloenzyme. That is going to be the functional part of functional enzyme. If you're missing that, if you don't have the cofactor involved, we call that the apo. Apo is like without. So apoenzyme is going to be the inactive form of that protein. It's not going to have the cofactor involved. Cofactors are chemically changed by the enzymatic reaction. So the coenzymes will be changed. The enzyme itself should not be changed, but the coenzymes will change. In order to complete the catalytic cycle, the coenzyme has to go back to its original state. So it has to revert back. So that one will be changed during the course of the reaction. We also have things called vitamins. Vitamins are things that your body's not going to make, so they're not synthesized by animals. You have to get them from your diet. So these are like essential minerals. They can also be used as coenzymes. All the vitamins that are used as coenzymes are going to be the water soluble ones. Fat soluble vitamins are not generally components of coenzymes. The water soluble ones will be. Um, and they're required in trace amounts in many animals. And the book kind of talked a little bit about the process of evolution. Through the process of evolution, we lost the ability to synthesize some of these vitamins. That's kind of interesting. And then here are examples of some of these vitamins. I probably won't test you on them, but you may have seen those. If you take vitamins regularly, you might see those on the package.